will go ahead and start the recording. Oh, and I will, okay, I will start the symposium. I will start by introducing Dr. Maria Pappas. I first want to thank you all to attend this last 2023 webinar. Again, if you know people who cannot attend and would like to see Dr. Pappas and other speakers' presentation, we have a YouTube channel where you can find this presentation later on and view it in a later time. So let's start with a little introduction of Dr. Maria Pappas. So she's an associate professor in entomology and acarology at the Department of Agricultural Development, and she's a member of the Laboratory of Agricultural Entomology and Zoology at Democritus University of Trace. She obtained a first degree in agricultural science, specifically in crop protection, and then a PhD in entomology from the Aristotle University of Thessaloniki in Greece. She worked as a postdoctoral researcher at the Universiteit Berlin in Germany and at the University of Amsterdam in the Netherlands. In 2014, she was awarded a Fulbright Award to perform a research at the University of California, Riverside. Her main research interests are in ecology of arthropods, pest and biocontrol agent, and in plant-herbivore interaction. Her current work is focused on biological control and tree trophic interaction between arthropods, plants, and microbes. She has published 46 papers and has more than 1,000 citations on topics related to herbivore-plant interaction, integrated pest management, and biological control. Dr. Pappas has coordinated and/or participated in 12 projects funded by the European Union, international and national bodies, and 38 projects funded by agro and biocontrol industries. She is also a member of the Gender Equality Committee of Democritus University of Trace and the vice head of the Department of Agricultural Development. So with that, you can start sharing your screen and we'll be happy
thank
to.
you. Thank you very much, Emily, for this presentation. Introduction. Okay. So I'm doing the full screen now. I think now you can see my
Yes.
first slide, right?
Yes.
Great. So thank you for this invitation. I'm very happy to be among you. It's a pleasure. And I'm going to start with a little bit of introduction of what we are doing in the lab, not of myself, because you have heard a lot from from Emily before. So today I'm going to talk about beneficial soil microbes and how we can use them against the key mite pests of tomatoes. So I decided, of course, to focus on mite pests, but I must say that during the last few years, we have been working in the lab with the effects of soil microbes against a series of different herbivores pests, not only mites. But for this presentation, I'm going to focus on mite pests. So here you can also see my email address if you twice, as I can see. So if you have any questions afterwards, you can contact me and we can discuss on any scientific matter that you that arise during my presentation. And for this talk today, I'm going to give you first a little bit of a theoretical background on the topic, but also the main part is to focus on our main outcomes of the experiments that we have performed the last few years. Through at least two different projects. So you can see their logos here. The first one is the BMOS project, which is a nationally funded project, and also the Intomed project, which is a Mediterranean funded project. I'm sure you're not aware of these projects, but yeah, these are the projects that have been funded, and we're very happy to do so in the last few years. Before I can start, just to show you where I'm located. So I'm located, our department is located at a small city called Orestiada. I think you can see my cursor also here. I'm showing the place where I live and work, which is near the borders of Turkey with Greece, and the north and eastern part of Greece. And here are some photos of our main buildings. And I'm a member of the Laboratory of Agricultural Entomology and Zoology, where we perform a series of different experiments, ranging from lab experiments with whole plants, also greenhouse experiments, and the last few years we have also organized a molecular ecology lab where we are very happy to start performing molecular analysis, and also to study chemicals, the chemical mechanisms involved in insect-plant interactions. So the main focus of our research is on insect and mite ecology study. And specifically, the last few years, we have focused on the plant-herbivore interactions. And our main goal is to identify or to help develop innovative tools that can be incorporated in biological pest control in order to substitute the toxic pesticides in the future. And through the funding by different organisms, by different funding agencies, we have been very happy the last few years to work on these research lines, and have been focused mainly on tomato from the crop that you can see here, but we have started also working on other solanaceous plants like pepper and eggplant. And here you can see the outline of my presentation. So first, as I said before, I'm going to start with a brief of a background on plant defense to herbivore, because this is very much related to what we are doing in our experiments for the effects of beneficial soil microbes against mite pests, herbivores in general, and specifically mite pests. And I also am going to focus a little bit on the priming, mechanism, and then to the beneficial organisms, specifically beneficial soil microbes as defense elicitors. And finally, I'm going to present to you some of our results, the results of our experiments that are related to the assessment of different beneficial soil microbes against above ground herbivores, specifically mite pests like spider mites and the tomato russet mite. So let's start with the background of plant defense to herbivory. Here you can see a tomato plant, a sole tomato plant, which is unfortunately facing a lot of different herbivores found above ground, like spider mites, whiteflies, aphids, and also moths, caterpillars, that have different feeding modes and belong to different feeding guilds, but they can also all attack the plant, tomato plant above ground. And this can also happen with plant pathogens above ground, but also with other parasitic organisms like plant parasitic nematodes found below ground. Fortunately for the plant, there's also beneficial organisms found above ground, like specialist predatory mites. So you can see here omnivores predators, zoophytophagous predators, and also parasitoids. That are all found above ground, but and this is of special interest to us, below ground we can also find beneficial soil microbe organisms like endophytes or plant growth promoting fungi, and also plant growth promoting bacteria. And this all modulate the ways the plants can respond to the attacks of herbivores found above ground, and also impact the behavior of their natural enemies. We know that plant defense against herbivory can be, there are two different types of plant defenses. The first one is direct defense, the first category, which includes compounds like toxins, antifidants, physical barriers, and also other plant traits that deter herbivory, meaning that they negatively affect the herbivore performance and also the behavior of the herbivores. And here you can see some examples of direct defenses, like nicotine or the glycosinolates, and also the famous tomato trichome, glandular trichomes, that are a very effective direct defense of tomato against herbivores. But also there's the second category of plant defense against herbivory, which is very also important and well known, the indirect defense, which includes the plant defensive traits that protect plants against herbivory by enhancing the attraction of the natural enemies of the herbivores, like parasitoids or predators. And there's one very important example shown here, which is the emission of plant volatile compounds by plants that are infested by spider mites, for example, by herbivores in general. And these compounds, this blend of compounds informs predators, natural enemies like predatory mites, like you can see here, which are attracted to these plants, and eventually they will suppress the populations of the herbivores best. This is the most well-known indirect defense responses of plants. But to be able to respond to a given herbivore species, plants, of course, needs first to perceive the presence of the herbivore. So here you can see a very nice figure. You should focus to the left part of the figure, where we can see the different herbivore species, again belonging to the different feeding gills. Here you can see also spider mite. Depending on the herbivore, the plant needs to recognize, perceive the presence of the herbivore. And this can be done by the so-called herbivore-associated molecular patterns, which can be compounds related to the presence of its herbivore and could be also specific to its herbivore. Like, for example, compounds that are related to the secretions of the herbivore, compounds found in the saliva of the different herbivores, or on the deposition fluids of its herbivore. And besides humps, the herbivore-associated molecular patterns, there's also the so-called damage-associated molecular patterns that you can see here for the pathogens, but the same holds also for the case of herbivores, which are not so specific to the presence of the herbivore, but are related to plant compounds, for example, that are released after the disruption of the plant tissue after the feeding by the herbivore. And these humps and dumps should then be perceived by the so-called PRRs, which are receptors, pattern recognition receptors, and then leads to pattern-triggered immunity. This is more or less a similar mechanism that we know also for what's happening to pathogens and the recognition of the pathogens by the plants. But unfortunately, we do not have so much information for what is happening with the perception and recognition of herbivores in that case. There's also the possibility that effectors, compounds related to the presence of herbivores, could also lead to pattern-triggered sensitivity. And finally, there's also the possibility that the so-called R proteins can perceive the effectors of the herbivores, and then again result to effector-triggered immunity. I'm not going too much in detail into this procedure, but just to highlight that it's important for the plants to understand, to perceive the presence of the herbivores, and that there's important mechanisms by which the plants can respond specifically to the different herbivore species. Also, it's important to note that these responses can be performed, can be induced locally, both locally but also systemically, not only on for example, on the same leaf where the herbivores are feeding, but also systemically in the plant, and also that there's important phytohormones like the desmonic acid, salicylic acid, and ethylene, but also the ABA, abscisic acid, that play important roles in the modulation of the different responses of the plants against herbivores. And finally, there's also the possibility for herbivores to defend themselves and to cope with plant defenses. And this can be done with a series of different mechanisms, like, for example, the suppression of plant defenses or behavioral adaptations, or other adaptations like the sequestration of the different compounds. So herbivores also have the ability to cope with plant defenses. They're not they can also defend themselves against the plants. Not all herbivores, but some can do. And a very important mechanism related to the induction of plant defenses, it's the so-called priming, which is, of course, the mechanism that you can understand that may be related with with plants that are prepared to defend themselves against herbivores. But what's the difference with the induction of plant defenses? I think this is important to highlight. And I wanted to do so with this figure, where you can see the response intensity of two different groups of plants. The first one, which is depicted with this blue line, relates to plants that are primed. These are the primed plants. And the gray line here depicts the plants that are induced plants. The induced plants after the attack is happening. For example, the attack by herbivore species or even a pathogen, for example, the inoculation by a pathogen. And I think it's clear that you can see here that the primed plants, the response intensity by the primed plants, it's happening, it's higher, of course, and that the primed plants seem to respond earlier to this attack as compared to the unprimed plant, but the induced plant. And it's very interesting to note that to be able to develop primed plants, we need some stimuli stimulus to act here in an earlier phase. And this stimuli can be a variety of different agents, like, for example, as you can see here, different bacteria, chemical stimuli, beneficial arthropods also, and beneficial microorganisms that they can act and then prepare the plants for the future attack. So this is important to note again, that beneficial organisms and chemical compounds that may be related to the presence of the beneficial organisms, besides abiotic cues and harmful organisms, can act as play the role of priming agents and can be used to prepare the plants to be able to respond faster and better to an attacker coming later on. Beneficial microbes, soil microbes in particular, that is also the focus of our work, is one very important category that includes many different strains and organisms, biological control agents actually, that could be used as the defense elicitors, as I explained before, through the priming mechanism and through the induction of plant defenses. And this is a figure I copied from a review paper that we had published back in 2020, that you may want to have a look. where you can see that the different organisms found below ground, that they affect the modulation of the different phytohormones. These are the main phytohormones that I mentioned before, and how this could affect all these organisms that are found above ground. And this, of course, can be herbivores and pathogens, but also their natural enemies, and also pollinators, of course. Through different mechanisms that can be included in, can be found both in the direct defense category, but also in the indirect defense category that I mentioned before. Particularly the beneficial soil microbes, meaning the microorganisms that can be found in the rhizosphere, are very interesting because they are known to improve plant growth and also antagonize pathogens. But also we know from the past, many years ago, have shown different scientists and different research groups that they are able to prime plants, the plant immunity against future attackers, and mainly the vast majority of the literature is about pathogens. But in the last few years, there are many research groups focusing also on the effects of beneficial soil microbes, specifically plant growth promoting fungi, the so-called PGPF, but also rhizobacteria, the PGPR, that have shown potential in protecting the plants from herbivores found above ground. And this seems to be related to the elicitation of systemic defense responses through the so-called ISR, induced systemic resistance. So this is an example showing how different, in this figure, how the inoculation of pepper plants, for example, with soil beneficial microbes can affect, first of all, the indirect defense response of a plant by the elicitation, by the alteration of the volatile blend emitted by this plant, and this may result in the increased attraction of predators to this plant, that then will result in an increased prey consumption. Here we can see a whitefly, but this can also happen to mites, against spider mites, for example. But also through the induction of direct defenses against the herbivore by the elicitation of defense responses. So we see the soil beneficial microbes to have a particular interest as vaccination agents. And this is the way we use them also in our experiments. We inoculate the plants with beneficial soil microbes, and then we see the effects, we study the effects on the herbivores found above ground and their natural enemies. So with this, we can, I think, start with case studies, which are the experiments, the description of the different experiments that we have done with different herbivores, focusing only to mite pests for this talk, and also the natural enemies of mites, to assess the effects of beneficial soil microbes against spider mites, specifically Tetranicus urticae, which is a very important pest, as you can all know, I think, infesting many different plant species and causing economic damage, not only to tomato, but in general to many important crops. But also Aculops coppersicae, which is also a very important key pest of tomato, which is not so easy to control, I must say. So these are the two mite pests that we are going to focus on in this talk. And what we hypothesized was that, of course, that these tomato pest interactions may be affected by the presence of the soil microbes by the induction of plant defense. According, of course, to the literature, specifically to the literature related to other herbivores, insects, but also to pathogens. And our experimental system consisted, of course, of tomato plant. The tomato plant specifically, you have used the cultivar Moneymaker, which is a well-known cultivar. For this cultivar, we also have important molecular tools to work on. So this is why this was selected, and also have plenty of literature related to this cultivar. And the two herbivore species, the Tetranicus urtica and Dactylops lycopersicii, and have assessed the effects of 16 different fungal and bacterial strains, including both lab and commercial ones. So we assess the effects, the plant-mediated effects of these strains to these two pests found above ground, infesting the plants above ground. Here you can see a table of the different strains that we have used. So there are many Trichoderma strains, one Pecilomycos strain, also Rhizophagus or Rhizoglomus strains, and Funeliformis. And there's also the Fusarium solinii strain K, which is a lab-owned strain by our collaborators. And you can see here the different concentrations that we have used, and also the information related to the commercial ones. The same holds for bacterial strains, mainly Bacillus strains, and also one Pseudomonas. So all of them consisting 16 different strains that we have used in 16 independent experiments against mainly the spider mites. So in our experiments, we followed a general methodology, the same methodology for each different strain. So we have started by sterilizing the pit that we have used to where we transplanted afterwards the young tomato plants, the Moneymaker tomato plants. And then after 2 days after transplantation, we inoculated the plants with different strains each time. And then after 3 weeks, we infested the plants with a certain number of spider mite females. Here you can see the 45 females and the different leaflets that we infested per plant. And then after 4 days, we counted the number of spider mites and the number of eggs found on each plant. And of course, we had all the different treatments, like, for example, plants that were only infested with spider mites, plants that were infested with spider mites that also inoculated with the microbe, to be able to see the differences between the different treatments. And we did also experiments with different commercial cultivars as well. We did not focus only on Moneymaker, and also assess the effects of the herbivores on the colonization efficiency of the promising microbes after the performance experiments. And also for another group of experiments, experimental plants, we also assessed the effects on plant growth parameters, because this is important, as you can understand, as we used soil microbes that are known, many of these are known to improve the plant growth parameters. And for another set of experiments and for specific promising microbial strains, we did some gene expression analysis. So this is what I'm going then to present the results of these experiments very briefly. For the experiments with the Dactylops lycopersicii, we followed the same protocol, but with a difference that we infested the plants with 40 individuals, and then counted the number of mites after 2 weeks, so for a longer time as compared to the experiments with the spider mites. So coming to the results for all these experiments, here you can see the results concerning the performance effects, the effects of the different microbial strains on spider mite performance. And on the first figure, you can see the reduction compared to the control of the different strains on the number of females found on each plant. And to the other figure, you can see the effects on egg oviposition, on the number of eggs found on each plant. And with red color, I think you may be not be able to read all the different species strain names. But what I wanted to show you here is that with the red color, you can see all the different strains that we have recorded. We found statistically significant differences as compared to the control. So we're happy to see that for all the different strains that we have used, there was a negative effect on the performance of spider mites, both in terms of female survival or the number of females found in the plants, and also on egg oviposition. Which was not the case for the effects on the plant growth parameters. So here you can see in general many different things, but I think you should focus on the fact that there were no significant differences of the different microbial strains on the dry and fresh weight of the stems. And this was also the case for the weight of the root system. At least for the time period that our experiments were running, which was not a long period, we did not find any positive or negative effect on of the microbial colonization and infestation with spider mites on these plant growth parameters, which might also be a good, of course, result. And also with experiments on the effects of the opposite effects of the herbivores on the microbial colonization. We're also happy to see that for the most promising strains that we tested, we did not do that for all the different strains, but for the promising ones, it seems that spider mites did not affect negatively or had no effect on the colonization of the microbes, which is a good sign. Of course, we need to do experiments, long-term experiments, to also see that after longer time period, but at least for now, it's a good sign that spider mites did not affect the colonization of the microbes found below ground. As regards now the effects on the different cultivars and whether there was a consistent effect, negative effect on spider mite survival and oviposition on the different commercial cultivars that you can see here. These are the names of the different commercial cultivars that we have used. You can see with blue color and the asterisks those microbial or fungal strains and those cultivars that were found to respond similarly to the moneymaker cultivar. So not for all the cases, but at least for most of the cases, we have seen negative effects on spider mite performance when also we used commercial cultivars in our experiments, which is also a good result because we wanted, of course, these negative effects to be found for commercial cultivars as well. And here you can see some of our results with the Aclops lycopersicki for different fungal strains, Trichoderma. This is the population reduction, percentage of the population reduction per plant for these mites. We can see that there's a significant effect, but also for the FSK and the Rhizophagus regularis QS69, which were found to be the most promising fungal strains against the tomato russet mite, found very important negative effects on the population of this mite pest as well. After 2 weeks, 2 weeks after we infested the plants with these mites. As I said before, for another set of experimental plants, we have also prepared the plants as I explained before, and then we also used these plants to harvest the leaf tissue and do gene expression analysis for these plants, because of course we thought that there might be the mechanism involved in these negative effects might be related to the induction of plant defenses. And for this, we have tested several marker genes that are shown here. The first ones are related to the JA, the jasmonic acid phytohormone. So there were JA marker genes and also SA marker genes, but also some marker genes that are related to the volatile emission, but also to the presence of the fungal strains of the fungi. So we prepared the plants as before, as I described before, and then harvested leaf tissue and did the qPCR to do the gene expression analysis. And this we did not only for the spider mite experiments, but also with the Aclups lycopersicii. And for this, have used three promising fungal strains that you can see here. The first one is the Fusarium solanum strain K. The second one is the Trichoderma harzianum T22, and the last one is the Esophagus irregulare QS69. The two of them are commercial strains. The first one is a lab-owned strain. Again, we prepared the plants as before, and then we tested a smaller set of marker genes for these experiments that are also related to the JA biosynthesis pathway and also the SA pathogenesis pathway. And coming to the results for the gene expression analysis, these are quite complex figures, with many colors. But I wanted you to focus for the different marker genes to those that are highlighted in red color. That you can see here that are all related to the JA biosynthetic pathway, and also to these two bars per panel, the green and the blue one. And the mention here that there's a significant interaction effect that was found for these marker genes. So there were at least 4 or 5 different marker genes, 4 different marker genes related to the JA biosynthetic pathway that were shown to be related to significant interaction effects. So when both the plants were infested with spider mites and inoculated with they were overexpressed and they were inoculated with a microbial strain. But also this was the case for the PR1A, which is the marker gene related to the SA biosynthetic pathway, depicting that both the JA and the SA pathways should be involved in the recorded performance results that we have presented before. As regards now the effects of on Aclups lycopersicii and the different marker genes, here you can see the results on the effects of inoculating plants with Fusarium solanum strain K, the lab-owned strain. And in red circles, you can see all these different marker genes that were found to be significantly expressed in response to the inoculation of the plants with the microbial strain and also the infestation with the Aclups lycopersicii. And these are mainly related to the JA pathway, but also related to, there's our marker genes related to relative to the volatile emission by the plants that are infested and inoculated with pest and the microbe respectively. And the same holds for Trichoderma harzianum T22, which is a fungal strain for the same marker genes. So you can see that we have found significant interaction effects only in the case of PI2C, a JA marker gene, and also PR1A, and SA marker gene. So overall, for these results, we can see that we found significant interaction effects for many different marker genes, but these were different depending on the combination of the different herbivore species, the mite pest, and the microbial strain. So differential expression depending on the combination of microbe and pest species. This is also the case for QS69, which is a mycorrhizal strain, a very important one and commercially available. Again, we have found different marker genes that were differentially expressed in this case as well as compared to the previous combinations. What we also did in our experiments, not only with spider mites that I'm going to show you the main results here, but also with other herbivores, is that we assessed the effects of inoculating the plants with the promising beneficial microbes on the differential emission of volatiles. How this can affect the volatile emission as compared to the plants that are not inoculated with the same microbe and infested or not with spider mites. And then we were interested to see how this differential volatile emission can affect the behavior of natural enemies. And for these experiments that I'm going to show now, we used as a natural enemy this optophagus predator here, which is called Macrolophus pygmaeus, which is a meteorite predator. It can also feed from the plant. And we did some olfactometer experiments to assess the effects of plant inoculation, tomato inoculation with Fusarium solani strain K for all the different combinations between plants that were infested with spider mites or not, inoculated with spider mites and infested with spider mites and inoculated with microbes, only inoculated with microbes, and also plants that were clean, not inoculated, not infested, also against the air. So there were, I think, 6 different combinations that you can see here. Some of our representative results for this combination of Macrolophus and spider mite infested plants that were also inoculated with FSK or not. So these are the results of the olfactometer experiments. What we need to see here and to comment on, I think, is mainly these 2 last bars. So you can see that plants that were only inoculated with FSK were more attractive to the predator that were from plants that were not inoculated with FSK, but also that when these plants were infested with spider mites and inoculated with FSK, again, they were more attractive than when the plants were only infested with spider mites. So it's clear that the inoculation of the tomato plants with FSK enhance the attractiveness of these plants, either these are infested or not with spider mites, to the predator. So it's a clear sign of the enhancement of indirect defense from lab experiments that we need, of course, to see at larger scale later on in the greenhouse. And for this, we wanted to see, to assess the synthesis of the volatile blend. So we did some headspace volatile trapping from the plants of the different treatments, and then analyzed the volatiles by means of GC-MS, mass chromatography, mass spectrometry. And we did PCA analysis, but I think I'm not going to focus here. We see here some kind of separation between the different treatments in the volatile, the different volatile compounds that we have recorded, which were at least 40 different compounds. But I wanted to focus on these figures, which show also for the different treatments, the 4 different treatments, plants infested with spider mites, plants inoculated with the fungal strain and infested with spider mites, clean plants that were not infested or inoculated, but also plants that were inoculated with a microbe, that we had identified 4 different compounds that you can see their names also here that showed significant interaction effects. The most popular one, of course, is methyl salicylate. For these compounds, but also other ones from the literature and from the other experiments that we have performed with the other microbial strains, we are now in the process of assessing the individual compounds for those that can be found commercially, can be purchased, for their effects on the behavior of the herbivores, of the mites, of other herbivore species like insects, for example, and also to their natural elements, doing choice experiments and performance and behavioral experiments. And these were some unknown, of course, compounds, another 4 unknown compounds that we are now in the process of understanding which are these compounds. And finally, I wanted to show to you 2 slides with the results from our greenhouse experiments. So from the performance experiments, we resulted in eventually in the identification of 2 different or 3 different, in some cases, strains, microbial strains. The first one is Trichoderma hertzianum T22. The second one is Bacillus amyloquefaciens QST 763, which is a bacterial strain, and also the mycorrhizal strain, the QS69, that I showed you some results before. And with these microbial strains, we have continued our experiments, not only for the molecular and the chemical mechanisms involved, but also we went then to the greenhouse to do some experiments, some longer term experiments that you can see the results here. So in these experiments, we infested the plants with spider mites, inoculated with its microbial strain, and also included another treatment when we also used a biological control agent. In this case, we have used also Macrolophus pygmaeus, because we wanted to see the compatibility of the different microbial strains with the biological control agent and how this can affect its performance in the long term. And we did the infestations or the inoculations, and then followed the populations of did the samplings for 6 weeks in the row. And what we can see here is the effects on the Tyrannicus urticae, the spider mites, eggs per plant, individuals per plant, and also the predator individuals per plant for the different treatments. Of course, in the case of the natural enemy, you can see the 2 different treatments, only the 2 treatments. And what we can say overall, both for Trichoderma hertzianum T22, but also for the case of Bacillus amyloquefaciens, the bacterial strain, which were both shown to be negatively affecting the spider mites in the lab, also the tomato acid mite, is that these results were again shown for the longer term experiments. And in relation to the presence of the natural enemies, the biological control agent, we must say that at least we did not see any negative effect on the populations of the natural enemy. And this could be the case, might be the case. Our hypothesis was that this is a possible scenario because the Macrolophus, the Sophistopagus predator, feeds also from the plant. So there might be possibility there for the microbial strains to also affect negatively Macrolophus pygmaeus, which was not shown at least for the 6 weeks that we did these greenhouse experiments, which is a good sign again. So our current work, because both these projects are now approaching to their end, but we still have a lot of things to work on. Like, for example, analyzing RNA-seq data for the most promising combinations of microbial strains and spider mites specifically, and also the tomato acid mite leaf samples. We also are working on assessing the volatile compounds and also other plant compounds that were found to be interesting against arthropods in general, but also spider mites are included in these different experiments. We see many opportunities from this kind of studies, besides identifying bacterial and fungal strains that could be used to inoculate the plants and prime them for a future attack by spider mites or Aculops lycopersicii. There's also many opportunities related to the identification of plant defense inducers like plant compounds, for example, peptides and the metabolites that could be identified and then used themselves individually, exogenously applied to make the plants, to prime the plants against future attackers, like plant protection tools, for example. There's also challenges, I must say, and the most important ones is the context dependency. You can see here that I have talked about different combinations of microbial strains and herbivore pests that resulted in the differential expression of marker genes. And also have seen that for the performance experiments between the different herbivore species that we have tested, not all the different strains were shown to be negatively affecting at the same extent the different herbivore pests. So the plant arthropod microbe combination specificity is a very important challenge that we need to take into account, and also that we need to go, of course, to the field because most of these experiments were lab experiments, and we were interested to see to identify the mechanism, to study the mechanisms involved in order to be able to identify promising compounds in the end that could be used solely as exogenous inducers. But in the end, we need to test all these under more realistic conditions in the greenhouse or in the field. And with this, I think I could finish. These are some of the people that have worked on these different projects that are approaching to their end, unfortunately, but we are happy enough to have two more the EcoBoost and Aster projects that we are going to continue this research, mainly in the direction of identifying and testing plant compounds like substitutes of like plant defense inducers and substitutes of the harmful synthetic pesticides that are needed to be, according to the EU Green Deal, to reduce. We need to reduce their use very, very soon. So with this, I think I can take questions if there's any, and I would like to thank you for your attention. I hope you can still hear me.
Thank you. It was really interesting. I'm gonna let people chime in and ask questions if they have some. Please unmute yourself.
So just stop sharing.
Yeah, if you want to, yeah.
Okay.
So that maybe we can see each other.
Great.
Okay.
There's a comment in the chat.
Very informative. Thank you. From Malik Abakar.
There's Livia also here, I can see. Hi, Livia.
I ask.
Yes.
Yeah, my name is Crispus.
Crispus, do you come from the lab of Nicole Van Damme or no?
Yeah, I did my PhD there.
Okay.
And now I moved to Liverpool. I work with Sharon. I don't know if you know Sharon.
Okay, yes, I know her by name.
Okay. So do you think if the density of the mites were different, either too many or too few, the results would be the same?
Yeah, what the selection of the different densities were done according to the literature first, because we wanted to assess other protocols that have been used before from other research groups that have been using the same densities. But I think you're right. Of course, many more spider mites could have resulted in different responses. But also we have tried to use a density that's more related to what is happening in under realistic conditions. Not so many, but not so low numbers of different spider mites.
Yeah, it's something we are trying to see with our plants
Okay.
here, because we also
Okay.
work with bacteria and in barley, and we
Yes.
don't know which density is enough to induce really strong defenses. So yeah, it's like
Yeah.
you have one insect and you have 10, they have
Yeah.
the same effect or different. So
Actually, the experimental, the methodology that I've used, the setup was more or less similar to what the group of marine content Livia is here, so she knows, I think, better. This was the experimental setup that we have used to infest with spider mites the different positions of the plant, and then to assess the performance of the spider mites. The only difference was that we also inoculated the plants with the microbial strains.
okay, thank you.
Thank you.
I think there is a question from Lydia.
Hi,
Hello,
Livia.
hi,
Hi.
hello everyone. Hello, Maria. It's good to see you
Hi.
again.
Yes, good to see you.
Yeah, nice presentation, very informative. And as you know, I love this system, this tomato mite interaction.
Yes.
And now with the microorganisms, you really did a good job researching and digging very deep into the plant defense system. And I was actually wondering whether did you see an effect which could be even positive as well, or maybe negative, in the plant fitness?
This is the question everybody has.
Really?
When I present this kind of results in the different conferences and so. Not yet. So we did not go to the crop performance yet. So we are planning to do so with microtome plants, because you know that the microtome have shorter cycles, so it's easier to work on. But I cannot say more for the moment, because we have not the results analyzed yet. So yeah. But of course, it's important to see that these microbes do not negatively affect the crop performance, or even better, to be in favor of the crop performance. I think they should be like that, because they are not only plant defense inducers, but they are also improving. They should be improving the plant growth. So I don't know, but it all depends also on the final the final results. We need to see the results.
Yes,
to analyze the
okay.
results.
Very
Yes.
nice. Yeah, because I'm also positive that, I don't know,
Yes.
it's like a feeling that I have. They could be mutualists, and then they
Yeah.
can benefit each other. That would be
Yeah,
very
but
interesting for
it
your
should
study.
also depend also, I think, on the combination between the herbivore species and the different strain. Because it will depend on whether the plant is becoming a better quality food. This is also a possibility for the herbivore, and then in the long term, we may also face better population, higher populations of the spider mites, for example. At least for the 6 weeks that we have tested, this was not shown. It was not the case. Yeah, but we need to see.
Okay, thank you very much.
Thank you, Livia.
Have a nice day. Bye.
Thank you, Alexandra.
Hey, it's a great presentation. I really enjoyed, and I'm happy to see all these things going on in groups, and bringing
Yes.
a lot of people together is fantastic. I do have a question regarding predators. So in the white tube experiments, you use an omnivore, but that they do have an interest on the plant, right? So I was wondering if you have tested predators or if you have a hypothesis of how you might see the predators responding, that they are not that interested on the plant.
You mean other predators, not the Zoophytophagous predators?
Exactly. So like predatory mites, that they target the prey, they don't necessarily feed on the plant.
Yeah, I didn't check, for example, Phytoscilus persimilis that might be expected, but we tested for another combination of herbivore pests, like for example, like whiteflies, combination with Amblyseius wirsky. And there the results were not so positive. So it was not, we did not see that the predator was actually attracted more to these plants. So it also depends on the natural enemy, of course. This is why I said in the last slide that the context dependency, it's a major challenge with this type of experiments and work in general. So when we cannot easily say that we are now in the process of applying the bacterial or the fungal strains in the field, because then we found a very important strain that could enhance the indirect defense or enhance the direct defenses of the plant. So it depends. Yeah, we have also only tested with Amblyseius wirsky and plants infested with whiteflies or not.
And in the case of the whiteflies, you saw similar trends and responses as with the spider mites, so inoculated plants?
No, this was not so promising. So we did not see. I must say that the whitefly infestation seems to have a stronger effect to the attractiveness of the plants to the predatory mite as compared to the inoculation of the plants with the microbe. So this was not so strong as we saw with Macrolophus, for example.
And any hypothesis as to why is this happening?
Maybe it's related to the fact that you said before that this was a Zoophytophagous predator, so it's its interest is much more related also to the plant itself. We have seen differences also between myrid predators, for example, Macrolophus pygmaeus and Nesidiacoris tenuis, and I think this is related to the ways the different species respond to the different volatile blends. I'm not sure. I don't think I can say for sure of this. It's related to the different species and the way they respond.
Thank you. Very interesting.
Thank you.
Right. I think I will close up this seminar and this webinar annual. I hope someone will take over because it was our last seminar for 2023. So we'll see in 2024 if we will have over a webinar. But thank you so much, Dr. Maria Pappas, for your presentation. And it was nice to hear about your research. And yeah, we hope to see you and hear about you later in the future.
Yeah, me too. Thank you very much for this invitation and for the very interesting talk. It was nice to see friends among you. Thank you very much.
Have a good evening. Bye.
Yeah, bye.